All right, so our focus is really a central point in all malignancy, and that is how do you control the cell cycle. To do that, there are a couple of reasons for doing that. For one, we just heard about the rate of mutation. If you can stop the cells, malignant cells from cycling, you automatically reduce the rate of mutation. In addition, I think by now we understand that the malignant cells and normal cells tend to be quite different in metabolism in many respects. So targeting the cell cycle has the possibility of actually distinguishing malignant cells from normal cells. I may not have time to get to that, but actually that's exactly what we found in terms of cancer cell metabolism. So let me just take you through, I purposely want to tell a simple story, but a linear story to give you an example of how we get from bench to the bedside. I'm sorry, let me go back. The focus of our work is really targeting the entry into the cell cycle from G0 to G1. That's where the cells grow and ready for replication, for division. And why do we do that? First of all, is that the CDK4 is one of the hallmark of cancer. It's literally overexpressed and never mutated in many, many solid tumors, including breast, including all blood cancers, and also the coupling, the biochemistry is very clear. But more importantly is that early in the 90s and early 2000, we knock out one of the inhibitor and realize that we no longer have normal plasma cells. So that means this is a very important step in controlling the terminal differentiation of plasma cells. And by inference, it probably is likely to be important for myeloma as well. So we fortunately have the privilege of working on a specific CDK4/6 inhibitor since the first day it became available. It's been about 8 years, and I'll take you through that. So what is the goal? The goal is really to develop a superior mechanism-based combination therapy using novel agents specifically targeting the cell cycle. Of course, we want to identify predictive biomarkers. And finally, we want to use functional genomic for patient and therapy stratification. And I think we have a glimpse of hope towards the end of the talk. Our approach is really from bench to the bedside, and we learn very humbly from what we see in the patients and back to the bench. And then we will use integrating novel reagent with basics research, clinical application at the team at Cornell. So this is the molecule. I think by now many of you already know about this. It has a new name called palbociclib. It's become available to us in the early 2005, and we did most of the early work. It's by far, so far, it's still the only specific CDK4/6 inhibitor, and it's reversible. It arrests cells in G1 in a very precise way, and this is an important point. I'll get back to that. As the therapy is orally bioavailable, it's low in toxicity, and this is the work done in my laboratory. We were the first to show that when we took primary myeloma tumor cells in the presence of stromal, these inhibitors stop the cell cycle and sensitize them to various agents, and then in the animal models. So having this as a background, then I want to take you forward to today. And all the red ones are the clinical trials that using this inhibitor that's been pioneered at Cornell. In particular, the single-agent mantle cell lymphoma represent the only published first in human trial using this agent. And I will take you also quickly about the consequences and the results. At the moment, we have completed a PD combination with bortezomib trial in myeloma, as well as the matching trial in mantle cell lymphoma. We are beginning to go into PD plus BTK inhibitor and PD plus lenalidomide. And it's the last one that I want to take you through. The important thing is that because it, as I mentioned, that CDK4 overexpression is also a hallmark of breast cancer. This molecule inhibitor is now designated as breakthrough therapy because in breast cancer, in combination with letrozole, it increases the progression-free survival from 7.6 to 26.1 months. In other words, more than tripling. So we now know for sure that this useful agent will be available in the near future. Okay, but we need to know the mechanism, and we need to know how to combine it with a cytotoxic partner, because no single agent can cure any disease. So first of all, I want to introduce you to the fact, despite the fact that myeloma has often been considered indolent disease, is not actually so. Even within a patient, it's very dynamic. And for example, we published this in 2006. This is just 4 representative patients at different stages. If you just focus on the blue, which is Ki67, and the green, which is BRDU, we label the cells with the BRDU for replication, you could tell that the different patients, stage 1, 3 relapse, and 3, they have different level of Ki67, representing different level of proliferation. And that's matched by the BRDU labeling, plus the other DNA replication markers, MCM7 and NCM2. So that led us, because the reagent is so specific, that led us to put forward this hypothesis, and so far it seems to be holding up quite nicely. The hypothesis is this, that because gene expression is coupled to the cell cycle in order to conserve the energy of the cells and the physiology, then we reason that if we could stop the cells very precisely in the middle of G1, and only the genes that are programmed here in the early G1 will be continuously expressed, whereas the genes that are programmed in late G1, SG2, and M will not have the chance to be expressed. And thereby, we are forcing an imbalance in gene expression. And the imbalance in gene expression will have physiologic consequences. And that's how we believe we can reprogram the cells. And this is an example, an early example, and we reproduce this again and again. And I will just take you through. The first, this is a replicating myeloma cell line, MM1S. You will see the same in many, many other cell lines in our hand. The first is asynchronous culture. The second is just barely arrest in G1. The third line, 24-hour, is prolonged G1 arrest. We call it PG1. I take you to the left. This is where no cell is in S phase. Then we remove the inhibitor, and the cells begin to come back to S phase, the beginning of S phase, and 18 hours later, it's entirely in S phase, and then it goes way back. So we look at the gene expression profile. The bottom is totally symmetric. This represents every gene that's in the cell cycle. In other words, all the genes in the cell cycle can be controlled by one single stroke of controlling the CDK4/6. It goes all the way down, and by qPCR, some of them are reduced 60 to 100-fold, but they come right back. And that's responsible for the ability for the cells to go back to the cell cycle. However, you have a block of genes that are stuck in the G1 phase. They could not keep up with the cell cycle. You also have a block of genes that's elevated, also stuck in the G1, and this is what we call imbalance in gene expression. Okay? So this then come back to the second part of hypothesis. If we now know that the cells can go synchronously, the cells will go forward, but the gene expression are not able to catch up. Would there be another point in the cell cycle that the tumor cells are exceptionally vulnerable? And yes, there is, from G1 to S transition point. And we utilize this to then design the schedule for clinical treatment, because we know this is happening in mice as well as in tissue culture and in primary cells in the presence of stroma. All right, so this is the first clinical trial, as I promised. It's mantle cell lymphoma. And in 17 patients, 16 are observable. These are pretty advanced, aggressive patients. And with the 125 mg, it seemed to be quite well tolerated, except for fatigue and neutropenia. But the surprise is that there's actually one complete remission over 2 years, 2 partial response, a deep partial response, and 5 stable disease over 1 year. So for a single cell cycle supposedly cytostatic, we actually have tumor shrinkage. We have to wonder, what does it do? And what it does is it's actually a clinical validation of the PG1 hypothesis, is that you perturb the balance of gene expression, you actually leads to cell death when it's treated for 3 weeks in a row. This is a 3-week cycle. So then I want to take you forward to the combination of lenalidomide. I think this is an exciting agent that has a potential of being used as a maintenance therapy for myeloma as well as for mantle cell lymphoma. So what is our goal? Our goal first is, can we sensitize the cells through inhibition of the cell cycle and increase the clinical efficacy at the lower dose? The other is, how do we know, can we use this as a way to understand how IMiD killing, because so far I don't think it's entirely clear how it works. And then if we can do that, can we actually identify biomarkers and forward this is for the future? Would we be able to improve maintenance therapy, control stem cell renewal, and control second primary malignancy? All these would be for the future, but I'll take you through the first 3. And this is just an example of what we can do. And for example, this is a primary myeloma cells we co-culture in the bone marrow stromal cells. And if you look, the only way that these cells are killed very rapidly is when they are put in combination. And that's to sensitize the cells for 6 hours through the cell cycle control, and then add the lenalidomide or pomalidomide in the presence of stromal. And it's time dependent and dose dependent. And here is a panel of the number we have done. For lenalidomide, 17 out of 21 primary myeloma cases are responsive. We haven't done enough for pomalidomide, but 3 out of 4 are responsive. And everything I'm talking about in the spirit of sharing, and none of it is yet published. Okay, so how does this work? First, we wanted to know if lenalidomide itself can actually inhibit the cell cycle, and yes, it does. If you look at the BRDU, it goes from 35 to 15, so it does. But it's not completely effective. It's only inhibit the cell cycle in some cells and not all. And the right is not important, except to say when we treated the cells with the CDK4 6 inhibitor, the PD033291, it overrides the cell cycle controlled by lenalidomide and arrest the cells in early G1. So this reagent pretty much take over the cell cycle control by any other reagent. So could it increase the killing? Yes, these are 2 cell lines. You see lenalidomide, the open pomalidomide, the open bar, and this is the cell cycle control. But when you had the 2 together, there's an additive or synergistic effect of killing, and we can lower the dose of killing. All right, so how does it kill? As we by now know, the 2 nice publications, that the CRB and the E3 ligase plays a role, as well as the IRF4. Does the synergy go through this pathway? Yes, if you look at the 2 cell lines, IRF4 is reduced by lenalidomide, but nearly completely disappear when you have the 2 agents together. In the other cell line, it's even more dramatic. And this is corresponding to a greater increase of the E3 ligase CRBN. Now, the regulation of IRF4 is actually pretty complex. It's both transcriptionally as well as post-transcriptional. This is just one of the steps. We do not know completely how it works, but we are getting there. So with that, you can see the cleavage of caspase-3, that's indication of death, is increased dramatically. And without, this is the schematics about how it works. So PD blocks the cells in prolonged G1 arrest, and we treat it with lenalidomide, it increases CRBN, leads to greater degradation of IRF4, and IRF4 is needed for myeloma survival, and that leads to cell death. So how do we find out how this works? We really do not want to second guess the mechanism. We decided to go through RNA sequencing to have unbiased global view of what are the changes in various ways of treatment. And we do this in primary myeloma cells in the presence of stroma. So let me just take you through quickly about what this method does. Many of you already have done it or know it already. First of all, it provides information about absolute RNA abundance. In other words, it's different from qPCR that it's dependent on the probes. This measures the total mRNA abundance. You can compare one gene to the other. And also the variant, where there's single nucleotide variation in dells, whether there is deletions and so forth. It goes using a 50 by 50 pair end RNA sequencing. And for every sample, we get 76 million reads. Obviously, this requires bioinformatics. This will then be aligned to the human genome and use software to call for reference variants. And this is the algorithm. Now, this is just for the fun of it. I will not go through it, except to call your attention. At Cornell, together with Chris Mason and Olivier Elemental, we have worked out a very detailed algorithm to look at the RNA sequencing in myeloma, AML, as well as mantle cell lymphoma. And actually, for myeloma, it's not trivial because these cells, the majority of the RNA is immunoglobulin heavy and light chains. So the results tend to be masked by the overabundance. We actually have to have ways of eliminating that in order to review the rest of the transcription program. So here, just to focus your attention on the fact that we can have differential gene expression, splicing over isoform polyadenylation. But I think it's also extremely important that we can actually identify and discover single nucleotide variant and use program to predict whether those variants are potentially damaging, whether they are synonymous and non-synonymous, and so forth. So another important thing is if we want to study samples at remission, or even when the therapy is working, there will not be very many cells to study. So we devise a way to bring down the need of the RNA as low as 10 nanogram. And this you can see from 10 to 1,000 nanogram is a complete linear curve. That gives us confidence we can trust the data we are looking at. So with this is an example. Then we ask, in the case when we have combination and there is synergistic killing, exactly what pathway is at work? And very quickly, we realize it's the interferon pathway. So let me take you through this. This actually does not require a huge amount of bioinformatics to review that. So you can see these are the interferon responsive transcription factor. If you focus on the last bar, that's where the two agents are together, cell cycle arrest and plus lenalidomide. So this is RSAD2, interferon 7, and this is important. This is TRAIL. That's a receptor, a TNF family receptor. So we begin to know better about how it's killed. It is quite specific, as you can see, the IRF4 RNA itself, not only protein, actually goes down in synergy. It's very specific because IRF7, this is normal cells, and this is myeloma. There's no change. IRF3, there's no change. But IRF7, you see a change, okay? So because in the interest of time, I will not go through all the details, except to show you now we begin to be able to expand this pathway, understand better. So IRF4 is a repressor of IRF7. When it's reduced, it allows the IRF7 to be deep repressed. IRF7 is a potent inducer of interferon, and interferon beta also go back to autocrine to regulate IRF7. So you establish a loop of IRF7 interferon to kill. And the killing is mediated by TRAIL, because when we knock out TRAIL, there's no killing. If we knock out IRF7, there is no killing, all right? So I think that at this point, we pretty much, we know more about this, if anybody's interested, that the way that these two agents work together is mediated through this axis, CRBN, IRF4, de-repression IRF7, induction IRF7, and TRAIL, okay? Is that the only pathway? No, as we went on, we realized the Foxhole transcription factor and the induction of reactive oxygen species is involved. So we are working really hard to ask how these two pathways cross in order to lead to the final killing. And I think that is going to be very important because in the future, we will have a clinical trial. We want to know as much as possible the potential molecular target in order to interrogate the patient samples. So is that only working in myeloma? No, this is a case of mantle cell lymphoma. We took 2 cell lines that are very resistant to lenalidomide killing. In one, one's called Marver, absolutely no killing. In the other one called JCO, and asked, can the cell cycle sensitize it? And yes, in one of them, JCO, we sensitize it, and it's the same mechanism. It's a synergistic loss of IL-4. In a Marver 1 cell line, it cannot sensitize, and there's no loss of IL-4. We have sequenced all these cell lines, and both at the DNA and the RNA level, and I think we have a hint of why this cell line is not responding. So the next step is pretty exciting. So in summary, lenalidomide, pomalidomide induce incomplete late G1 arrest, and this is independent of RB. I think this is important because everything I tell you about interfering the cell cycle with the CDK4 inhibitor is RB dependent. Induction of prolonged G1 arrest can, by this inhibitor, can override cell cycle arrest induced by IMiD. And the prolonged G1 arrest reprogram cells for IMiD killing through a drastic reduction of IL-4. And this is through the interferon pathway mediated by IL-7 and TREL killing. So much for the work on the bench. What we have shown you is that the cell cycle can enhance IMiD killing, and at the lower dose. I didn't explain that, but that's the case. We learned something about the mechanism already. Do we know anything about the biomarker? I'll show you a tantalizing result. And then maintenance therapy, these we will address in an upcoming clinical trial. That's the protocol, and that will be headed by Dr. Thomas Mark and Dr. Nisvitsky in close collaboration. We designed this protocol together. We will sensitize the cells first with a CDK4/6 inhibitor, and then afterwards add the lenalidomide to the mix and keep it together. So I mentioned that we wanted to know about the biomarker, and I also mentioned we have completed a clinical trial with PD in combination with bortezomib, and we know in the myeloma field that patients' treatment is really either way or one way or the other. So is there any biomarker that we can use to predict what might be a better combination? And I am not at liberty to tell you all the results because the clinical trial paper is being written up, but we did the same thing with RNA sequencing and DNA sequencing. And we actually begin to see biomarkers. For example, these are 3 genes, is a panel. In the patient are the responding patient, just an example of them. This is normal cells. It's absolutely not expressed. It's not low, it's just 0. In all the non-responding patients, every patient expresses gene. At the same time, there are 2 other genes that's only expressed in normal cells and the responding patient and absolutely not expressed in the non-responding patients. And this is just a sample of the signatures that we pull out in combination with bortezomib. So the question is the expression this interfere with lenalidomide response and with the PD reprogramming? And we do know the answer, and that is no. So we now begin to see a way of looking for biomarkers that can distinguishing the 2 combinations, and we're obviously very, very excited about this possibility. And hopefully next year I can show you the whole panel about these signatures. And these are coming from patients treated by this drug. It's not ex vivo or anything, but of course the lenalidomide we'll find out after the clinical trial. So in summary, we have a hypothesis, and it seemed to be correct. it forces imbalance in gene expression, and it induces redox stress. I did not have time to talk about it, but I will be happy to share. And so far, we have combination therapy. We have done this with IMiDs, with a PI3 inhibitor, with ibrutinib BTK, and that's a clinical trial in mantle cell that should begin soon. It also that it distinguishes the matching the partners. For the S-phase entry, bortezomib, calcifimab, and cytarabine is better. So we also have a feeling about how to schedule the combination. And our way of finally understand the rest of the meaning of everything we do is actually to do genome-based patient and therapy stratification based on the unbiased genomic and expression analysis. And this is our team. We are very blessed to have a very cooperative team. The work I presented today is mainly done by Xianggao Wang, and also Maurizio Di Liberto, but of course we have the help from everyone in the clinic, the myeloma team, the mantle cell lymphoma team, the pathologists and the Genomic Institute, and the Celgene, Pfizer, Milan is supplying the reagent. And I didn't have time to talk about it. We also did a esoteric screen to further understand what genes are absolutely needed for bortezomib and calcifimab killing. And we have identified 3 candidate genes which actually match the clinical data. That's the next talk. Thank you very much.